Okay, so now I have a large string set. I can do cool stuff now. For instance, let's assume that I want to calculate the GC content of each of the promoters. Yeah? I can use the letter frequency function for that. You can find the explanation in the vignette. So the letter frequency allows you to calculate the frequency of letters in the sequences. When I scroll down a bit, you see that it's a collection of multiple functions that all perform similar functionality. You can see here the different arguments that you can set for this function. So what is X? What is the input? The input should be a string. Can be DNA, RNA, protein, whatever string, or the input can also be a string set. So it can work directly on a string set. The letters argument, the second argument, defines the letters that you want to find in these sequences. So you can look for 1. You can look for 2, whatever you want. So if you define 2 letters, he will see it as an OR, yeah? Except when OR is set to 0. Then he's going to keep them separately. When OR is not equal to 0, he will look both at G, so he will count how many G's there are in the promoter, he will count how many C's there are in the promoter, and then he will add these counts. So you get a total count for C's and G's. As prop, if you set it to true, he will not give you the actual counts, but he will transform them into probabilities, into frequencies, which is in most cases what you want. So what I'm doing here is in the string set, I'm going to search in each promoter how many C's and G's there are, and then I'm going to convert that number into a frequency. So when I run this, he generates a matrix with the results. Now, typically when you calculate this, you want to visualize this, you want to plot this. We do plotting with ggplot2. ggplot2 wants a data frame as an input, so we have to convert this matrix into a data frame. That's what we are doing here. So I convert it into a data frame and then I take a look. What does it look like? So it will give me for each of the promoter sequences, it will give me the percentage, the frequency of the GCs. Now, what is annoying? The name here of this column. It will give you a lot of trouble when you are going to use that name as a name in your code because of the OR sign here. So R will literally interpret this line as an OR, and that will give you trouble. So when you see this OR appearing in column names, you convert it into something that does not contain an OR. So that's what we are doing here. We are converting the name of this column into GC, simply GC. So when I run this and I take another look, you see that he has changed the name. And now I can make a histogram. So I have to load the ggplot package. I'm going to plot the data from the GC data frame, so from this data frame. What am I going to put along the x-axis? Well, the column called GC, and then I add a histogram geom. So when I run this, I get a histogram of all the GC frequencies in all the promoters from human.